Dr. Karstens is the mentor of both of them. I'm going to let her be the MC, and that way I can turn off my camera and eat my lunch, but I will be listening and look forward to the talks. So it's all yours, Lisa.
Thank you. I'm very excited to have Lily and Palmer, two interns that were working with me over the summer, tell you a little bit about their projects. You'll see their projects are quite different from each other, and that's based on their primary interests and also the needs of the research that goes on in my lab, which primarily uses a lot of data science and bioinformatics to try to understand bacteria in the bladder and how it relates to women's health. With that, I'll let Lily say a brief intro about herself, and she will tell you about some of the metadata work that she's been focusing on this summer.
Hi, everyone. I'm Lily, and I'm an undergraduate at the University of Southern California. I'll go ahead and share my screen. All right. So my presentation is going to be on the challenges and solutions for microbiome metadata cleaning and clinical research. For an outline of our presentation, I'll talk a little bit about overactive bladder, or OAB, since the metadata that I worked with was collected as part of the OAB study. I'll go over what metadata is and the importance of metadata, then walk through a sample metadata cleaning process and talk about some takeaways and future steps. So overactive bladder syndrome is characterized by symptoms of urinary urgency, urinary frequency, nighttime voiding, and urgency urinary incontinence, which is having a strong urge followed by immediate leakage of a large volume of urine. It affects around 19% of women over the age of 44. And the diagram to the right shows a basic illustration of what occurs with this disorder, but the pathophysiology is not fully understood. So recent studies have found that overactive bladder may be connected to interactions of bacteria in the bladder. So this overactive bladder study aims to identify what kinds of bacteria are found in the bladder and if there are any associations with symptoms of overactive bladder. The basic procedure involved collecting some urine samples via a catheter system. Then they sequenced the 16S rRNA genes from these samples, and the 16S rRNA gene is frequently used in urinary microbiome studies. Then after a lot of processing steps, they were able to identify what bacteria are identified by these gene sequences. And with that information, you can do analyses like these, where you compare the relative abundance of different types of bacteria in each of your samples. So in addition to collecting the gene sequencing data, you also want to collect metadata. And this is data that describes and gives information about other data. It's really important in sequencing projects because large volumes of sequencing data require context, such as where do the sequences come from, how are they processed, how are they produced, in order to understand and then analyze them. Particularly with microbiome research, metadata is important since this type of research aims to identify the interactions between the host and the microbiome. So having information about the host is really important to provide that context. And lastly, metadata helps to increase the reproducibility and facilitates data sharing and reuse of your work, since sequencing data is often banked in repositories so that it can be accessed by other researchers or yourself in the future. There are a few different types of metadata. What I'm focusing on today is going to be sample metadata. So in human subjects research, that would be information about the host, such as their age, their gender, what medications they're on, what pre-existing health conditions they may have. There's also information about the sample itself. So what type of sample is it? Is it a blood or a urine sample? And then other forms of metadata are preparation, data processing, and feature metadata, because there are a lot of different methods that can be used for processing your data sequences, or different databases that you can use to identify what bacteria those sequences align with. So having that information provided is also really important. So for the OAB study, the clinical metadata that was collected included a 3-day bladder diary. And this is an example of what an entry of one of those diary days would look like. So you would have your fluid intake, what type of fluid, if you had any leaks or accidents, if you urinated in the toilet, your urgency, severity, and any activities that you were doing when you leaked or had an urge. Other clinical metadata included information about the study visit, such as the amount of urine that was collected. There's also information about the sample. So that could be the results of a urinalysis, like shown on the right. We also collected the subject's medical and social history. That would include their pre-existing health conditions and other information about their medications and treatments. And in addition, there were some questionnaires about their quality of life, their stress and anxiety, and these all help to evaluate the severity of their overactive bladder symptoms. So there are a couple issues that are associated with collecting clinical metadata. The first is that it can be prone to entry errors since everything is manually entered by research personnel. And the second is that it can be disorganized or non-informative when it is first entered. To resolve these issues would require some data verification and cleaning processes in order to make the data more analysis friendly or tidy. So here we have an example of data that is untidy. This is what you might get when you initially enter the data in. And after, you would want to process this data to look something more like this, which is be a lot more analysis friendly. So the basic guidelines for making tidy data is having one observation per row, one variable per column, and then one measurement per the cell. Looking more specifically at the changes between these two tables, we can look at the species column first. So we had either Homo sapiens or human were entered for the species column in this data table, but we want the same meaning to have the same data value. So we changed both of them to Homo sapiens. For the date column, there are a lot of different formats for date. So we changed that to this, which is a standard format, so that it can be all the same in the cleaner version of the table. In the group column, we had some differences with either capitalization or abbreviations. So similarly to the other two columns, we made that to be a same value for the same meaning. Next is the location column, and you can see the column name for this one was capitalized while all the others were not. So you also want your column names to have the same format. So we had that be lowercase. And then for the values, this one was in city-state format, and this one was for OHSU. So we also changed that to both be in city-state format. The last column, you can see in the untidy version, there are two values in one column. So you'll want to split that up so they have their own columns. And then I also moved the units into the column name so that the values are just a numeric value, which will make it much easier if you want to do any calculations. And for the units, we also changed them so that they would match, since originally the units were different for the two rows. So that's for helping to kind of tidy up the data. And then the process for resolving entry errors would involve first performing a double data entry. So that would mean having two different research personnel both enter the data into a data set, so that you have two data sets. You would then identify any differences between the two data sets so that you can verify and resolve those differences, and that will help to minimize any entry errors. So the step that I focused on was the second one of identifying the differences between the two data sets. I used a couple R packages to help with this. The first is compare.df. It was downloaded from GitHub and created by Alex and Joseph. This contains a single function, compare_df, that will create a comparison objects from two data frames. The second package that works in conjunction with this one is HTML table, and it will create a table that highlights the differences between the two data sets. So prior to comparing the two data sets and using the R packages, I did some processing of the raw data. So this is an example of raw bladder diary data. And the first thing I did was help improve human readability. So I added labels to all the column names so that the data is more understandable and easier to work with. Then you want to ensure that the rows and the columns match between the two entered data sets so that you can do the comparison. So this is a subset of the initial entry, and this is a subset of the second entry. So here we have 5 variables that are shown, and then it's for 3 different subjects. So looking at these two, there were 2 main differences. The first was that all the column names in the second entry had an extra _2. And this is because when we do the double data entry and REDCap, it requires different variable names for each value. So we had to make the variable names for the second entry to be different from the first. But when we're doing this comparison, we want them to be the same. So in R, I was able to remove these extra _2 from the second entry column names. The second issue was that a few of the columns differed in their ordering. So this is for the day 3 data. And what I did was I looked at days 1 and 2, and the order of the accuracy, notes, and average intake columns matched the initial entry in this order, while the second entry was different. So I took the second data set and I reordered those 3 columns so that they would all match. And after that was done, now we have the 2 data sets where the rows and the columns all matched in their order and their names. So then we can proceed to creating the comparison table. So first we have that compare.df function here. The first argument is going to be the initial entry data set. The second will be the second entry data set. This value will be the variable that you want to group the columns by. So I wanted to group them by the record ID. And the last one is where you can put any columns that you want to exclude. So we wanted to exclude the notes column for the example that was shown, since that's a freehand column and it's going to be different between the data sets. Then you can run the create output table function on this created comparison object, and you will end up with something that looks like this. So on the left here would be the record IDs, and then these 2 rows would be for one record ID, where the top or the green row would be what is entered in the initial entry, and the bottom would be for the second entry. And then any of the values that are colored green are going to be unique to the first entry, and if it's colored red, it's going to be unique to the second entry. So this just helps to highlight the differences between the 2. And then from this table, you want to determine which differences actually require verification. So this is going to be different based on what data you're working with. But with the bladder diary data, the average values are the most important to have matched between the 2, because those are the values that are actually going to be looked at in downstream analysis. So for example, in the first 2 rows, there's one average column here, and the averages were different between the two entered data sets. So those might be record IDs that you would want to go back and verify. But the third one, there was one difference in this column here, but the average value and other average values that aren't shown in this slide all match between the two data sets. So this wouldn't be a record ID that you would have to go back and verify. So after identifying which ones require verification, you would then go and actually review those record IDs. Then we are working on resolving those identified errors, and I did both the bladder diary and study visit information data sets. And then after resolving those, you want to repeat the process just to make sure that everything that needs to be resolved has been resolved. And doing this in R is very convenient because then you already have the scripts written and you can run the data sets through that same script. Then once everything has been verified and cleaned, you can proceed to your analysis. And that covers the project that I did with the metadata cleaning. Some other takeaways from this internship have been learning how to utilize R for data cleaning. I've learned about the importance of documentation. All my work has been done in R and R Markdown files where you can write both your code and then also plain text to explain what you're doing and why you're doing it. I have also learned about how the collection of metadata plays an important role in microbiome research. And there's a lot of work being done right now in the Carson's lab and other labs and organizations to create metadata standards so that all researchers can collect and report the necessary metadata with their research, and that it can be more reproducible and reusable. And lastly, that studying the urinary microbiome poses many unique challenges. Since the urinary microbiome is a relatively new field, and there are a lot of questions still about what are the best methods to use and issues that are yet to be resolved that Palmer will also talk about in her presentation. And lastly, just some acknowledgements. Thank you to my mentors, Dr. Carsons and Dr. Gordine, the National Institute of Diabetes and Digestive and Kidney Diseases, who helped to fund the study, and of course the OHSU Department of Medical Informatics and Clinical Epidemiology for this internship experience. And thank you all for listening.
Thanks. Can we ask some questions?
Yeah.
Great. Very nice talk, and you exemplify actually a problem that we know is very challenging in our field, which is how we go from data that's collected in the real world, often for other purposes, and then we want to do research with it. And we run into these problems that the data is not standardized, it's in different formats. Before you actually got to the last slide about the next steps, I was going to ask, but I think maybe it's still a pertinent question. You know, how confident were you that, you know, for example, when you had height and weight in the same cell, that you parsed out those numbers properly, so that once you got into a more tidy data form, that you could feel confident that the underlying, you know, all the transformations you had done were correct?
Yeah. So for the example, that was just like an example data set that I made for the presentation, but it wasn't with the actual data. But for just cleaning the data in general, I guess just running it multiple times or going over and checking every time before you keep going. Or also, since we had the two different data sets, to run it on both and then be able to compare and make sure both were cleaned and tidy.
Mhm. Okay, I mean the takeaway point, is that we oftentimes have challenges with data sets that we reuse for other purposes. Not that we shouldn't do it, but we always need to kind of keep in mind where the data came from and how it might need to be cleaned. Thanks.
Are there any other questions for Lily? If not, we can move on to Palmer, who is going to discuss some of the work that she's been doing over the summer with quantitative microbiome profiling. So more focusing on the sequencing side of data and trying to improve the information we can get out of it.
All right, can you guys hear me okay? Hopefully. Okay, great. Let me get my presentation going. All right. All right, like Lisa said, my name is Palmer, and I'm a senior undergraduate majoring in math and biology at Lewis and Clark College here in Portland. And I was also in the Karsten's lab with Lily, but I was working in the quantitative microbiome profiling project, working on detecting bias. But before we get into that, I thought it would be beneficial to just talk a little bit about the human microbiome and bacteria. So bacteria, viruses, and archaea live all over our bodies, and most of the time they help our bodies function properly, exchanging materials and helping our systems. But when a microbiome's diversity is skewed or there's an abnormally large presence of one type of bacteria, things can sort of start to go wrong. And so the microbiomes in our body have different levels of diversity. So for example, our urinary microbiome has a relatively low microbial biomass, whereas the gut is considered to be high microbial biomass. And so there are two ways that we can look at and study these microbes. The first is culture-based, and the second, which we'll be focusing on, is culture-independent. And so this involves metagenomic or shotgun sequencing, which aims to sequence all of the microbial DNA in a sample. And the other type is marker gene sequencing. And so we're going to focus on marker gene sequencing because it's the method that was used in this project. And so marker gene sequencing works to just sequence a specific region of the microbe's DNA to determine what type of bacteria it is. And so this is often done using the 16S rRNA gene that is specific to bacteria. So this gene consists of multiple hypervariable regions. In this picture, you can kind of see them, that you can essentially search for to determine what type of bacteria it is. And so it's kind of, you can think of it as like we use fingerprints to identify people. We can look at a 16S rRNA gene as a bacteria's fingerprint to help us figure out what type of bacteria it is. And so this method has been verified by the Human Microbiome Project as well as the Earth Microbiome Project. And so, yeah, marker gene sequencing takes biological material and works to output the corresponding bacterial profile of that microbiome. So it increases our ability to measure biological communities and, for example, to understand diseases that occur in the gut, the bladder, the vagina. These are all places where we have microbiomes. And so, like in this picture, you essentially take a sample of bacteria, and in this sample we have 3 purple bacteria, 2 orange, and 1 teal. And we put them through the sequencing, and we get out how many of them there are, or their relative abundance in most cases. But like in this picture, you'll see that it misrecorded. It said there were 4 orange ones, 1 purple, and 3 teal. Bacterial profiles that go through sequencing are often biased. So that means that some bacteria are more easily detected than others. which is why it picked up the orange one more than the purple one, even though the purple had higher abundance. And so my portion of this project focused on a recently developed mathematical model for estimating this bias and applying it to the mock microbial dilution series, which I'll talk about in a little bit. And so McLaren, Willis, and Callahan are the authors that proposed and wrote this model. And so it looks at how bias distorts bacterial compositions measured by marker gene and metagenomic sequencing. And so this model is represented in a package in R, which is a statistical computing language called Metacal. And so R has lots of different packages that can help us make plots and do all sorts of things, but this package worked specifically on detecting bias. And so, excuse me, like I said, this package works on getting quantitative bias estimations and then using those bias estimations to calibrate our observed abundances so that they are closer to the actual abundances. And so, for example, let's say you sequenced a microbiome and this was your sort of like, this is your data. And so this staircase that's sort of like behind the points is what you know is actually in your sample. But when you put your sample through sequencing, this is what it recorded. Like the points in color are what it said the abundance of each bacteria was. And so the Metacal package takes that and it makes it more like this. So each of these points are now much more accurate and precise to the actual composition of the sample. And so we know how much bias is impacting each bacteria within the sample. And so I'm going to shift lenses here a bit and talk about the project in the Carson's lab that I was a part of and discuss how all this is tied together afterwards. So one of the projects that the Carson's lab is working on right now is quantitative microbiome profiling, which means obtaining the counts or absolute abundances of the microbes in a sample. And so specifically, the Carson lab works on microbiomes that have low microbial biomass, like I mentioned earlier. And so my project within this was to look at how bias impacts these low microbial biomass samples. And in order to see how bias impacts microbiomes of varying biomass, the Carson's lab and Kate Bowie created a mock microbial dilution series. And so there are 9 levels in this mock microbial dilution series, starting at undiluted, which is pretty much full concentration. So that could represent like our gut, which has a relatively high biomass, whereas these lower or more diluted samples could represent like the urinary microbiome that is lower microbial biomass. And so an important thing to note is that the mock microbial series has 8 different bacteria in it, and they are all of equal proportions. Those are the actual abundances, they're 1 to 1. And so this is what the dilution series looks like when we plot it. As we go along towards the more diluted samples, we get more contaminants, and obviously the data or the abundances change. And so another part of the quantitative microbiome profiling project is investigating the role that contaminants play in high versus low microbial biomass samples. And so it was sort of interesting to see how my project was impacted by these contaminants. And so just to sort of sum up this intro part, the question that I aimed to focus on for my project was, how well does the Metacal approach assess bias on samples of varying biomass and the mock microbial dilution series? So sort of putting together this bias calibration aspect and this dilution series. And so the first step that the Metacal package proposed was to sort of figure out what what does our data look like? What is in these samples? And so what I did to do this, or to get a feel for it, I guess, was to create this tile plot. And so the bacteria are on the y-axis, while the dilution sample is on the x-axis on the bottom there. And so D0, this is our undiluted sample. And then D8 is our most diluted sample. And so the tiles are colored corresponding to their relative abundance. And so dark blue tiles represent high abundance, whereas this yellow represents low abundance. And so because the mock microbial dilution series is 1-to-1 for all 8 bacteria, if we had a flawless sequencing event in which there wasn't any bias, then we would expect to see all of these sort of the same color, sort of this like mid bluish green. But our sequencing picked up these bacteria more than it picked up these bacteria. And so there is some bias at play. And the other thing to note is that the top row is this other type of bacteria. And so this represents all of the bacteria that we observed that were not the 8 known bacteria in the sample. So in other words, these are our contaminants. And so I think it's interesting to notice that like as you progress down the dilution series, the abundance of this other or contaminants category increases. And that is sort of what we would expect because low microbial biomass samples typically have more contaminants just because there's less stuff in them to begin with. And so that's just something to point out with this tile plot. I also created a box plot to show the quantitative microbial biomass data abundance and variability just a bit differently. And so you can see sort of the same trends. We have these were observed at a higher abundance than they actually are present. And then we have those that are observed at a lower abundance. And then this other category has some variability in it. And so using the Metacal package, the next step that I took was looking at the dilution series and visually assessing how much bias there was. With the tile plot and the box plot, we know that there's some bias in play, but we don't really know how much. And so to do this, I plotted the observed abundances on the y-axis and the actual abundances on the x-axis. And so, and then they're colored by the dilution series. And so this gray line is what is where the sample should lie if there isn't any bias. So that's like our perfect sample abundance right there. And then the other thing to note is that these points do appear in sort of a column because the actual abundances are equal for all bacteria because it's 1-to-1 for every sample. But you can see how some bacteria are observed at or some dilution series, the samples in the dilution series have higher than actual abundance observed for the bacteria in them, and some have lower. And so this gives us sort of a feel for how much bias. And then another way that I thought to look at this was maybe if we look at each bacteria, we can have a better sense because the overall graph shows that there is bias, but not per bacteria. And so each of these squares is a different bacteria that is present in the mock microbial dilution series, the 8 recorded bacteria. And then the colors vary based off of which dilution series they're a part of, like in the last plot. And so, for example, if we look at lactobacillus, we see that throughout the dilution series, lactobacillus was observed at a higher abundance than what was actually present in every sample. But with Staphylococcus, all of the dilution series samples are observed lower than what was actually there. And so the yellow dot in the middle of these plots is the unbiased or expected observation. So that's what we would want to see if there was no bias in play. And so it definitely does pick up so far, the Metacal package, on how much bias is in play with each bacteria. And so the next step was to sort of, we know there's bias, we know which bacteria are experiencing bias. How much? What is our quantitative bias estimation? And so Metacal has a function for this. It's called estimate bias. And so you put in the actual known abundances of your bacteria and the observed abundances, and it outputs the bias. And so we can kind of see the same trends here. These top three are the ones that were observed in like the dark blue in the tile plot. So those are observed at a higher than actual abundance, and then it just declines from then on. And so the table is helpful for analysis, and we can use these estimates later, but I think it is actually more helpful to see these plotted. And so this is just the same data, and you can kind of see which ones have more bias and which ones have less bias, but still some bias in play. And this gray line up the middle is the like perfect no bias line here. And so yeah, so it's just, I think it's kind of interesting to look at. And now we know how much bias is in play for each bacteria. And so the next step going forward would be to sort of calibrate our observed abundances. So using our bias estimations, Metacal has a feature where, or a function where it inputs the observed and the bias, and then it outputs the calibrated amount. So it essentially adjusts for the bias that is present. And so here, so this, I didn't get to this. My computer crashed yesterday. It was very damaging to my progress on this project. But going forward, it would be useful to do this. And so this bacteria is, or I guess they're fungus. It's from the tutorial for the Metacal project. And so these were their observed, and then these are their actual. And so this calibrated is using the observed and the biases, and you can see how much closer the proportions of the bacteria are to the actual after we've calibrated. And so I also want to talk a little bit about limitations. And so one of the main things that we found to be limiting with this package is that in some instances, you can't develop a control sample that covers the range of bacteria present in the community that you're drawing samples from. And so the authors of the package and the paper mention this limitation. They say it might be useful to do a partial calibration when abundances are known for like a key bacteria that's in the community, but they don't provide an example, and there aren't a ton of functions for that within the package. And so with the quantitative microbiome profiling data, we knew that all the samples were one-to-one. We knew the abundances relative of each bacteria. But if we were to pull a sample from a human subject, we're not going to know exactly what bacteria is there. We could know maybe one or two that we could expect, but we don't know their abundances. And so that's definitely a limitation that maybe this isn't super applicable to real world samples. And the other limitation that we found is that contamination in low biomass samples isn't super talked about. In this project, I just lumped all of them into this other category, but they take up a lot of these samples at low microbial biomass in this dilution series. And so I think it would be really interesting to see how contamination impacts bias, and if there were functions for that, that would also be useful. And then I also just wanted to touch base on sort of my takeaways for the internship. Going into this, I didn't know anything about the microbiome. I didn't know really how to use R, which is a programming language. I didn't know that bias even existed. And so I really had it was a really eye-opening experience for me. And learning about marker gene sequencing, all of the implications to do with that, how to plot images and figures in R, is definitely something that I'm going to use, hopefully for the rest of my life. And yeah, the whole contaminants situation is really problematic and a big issue. Same with bias. And so learning about that was really interesting. And then I wanted to just thank the OHSU DMICE whole department, the mentors and the staff. You guys have made this experience really seamless from a remote perspective, which was awesome. Like I felt really included all the time, which was great because yeah, being remote can be really hard sometimes. And so I also just wanted to thank Dr. Lisa Karstens and Kate Bowie, who are my mentors for this, and the entire Karstens lab helped out a lot. They made it a really great online community. So it was really enjoyable out there. And then I just have my resources. So any questions?
Great. I actually don't have a question, but I actually did want to mention that the way that Palmer learned about our program was through the AMIA First Look program. AMIA, for those who don't know, is the American Medical Informatics Association, which has this program that focuses on women pursuing graduate study and careers in informatics. And somehow, even though Palmer is going to school, what, about 3 miles or 5 miles away from OHSU, learned about our program through the First Look program. In normal times, they actually fund the travel for some number of college women who apply and get accepted. Maybe at some point in the future, and probably not this year, the program will go back to being in person. It was virtual last year, so we got to meet her that way, and I'm glad this worked out and that she got to learn a lot and contribute to Dr. Karsten's research.
So I have a question, Palmer, if I'm allowed to ask. So I thought it was really interesting with your bias estimates. It looked like some of the bacteria actually had more spread than others. Do you know why? Or like how would that be adjusted for if you're trying to correct bias? It seems like some of them, like you'd actually have to have a I don't know if you'd be able to correct for the bias and like say lactobacillus if you go to the plot that had the that showed how different it was from the expected. Yeah, here.
Yeah.
Like lactobacillus has a huge spread of like the amount of difference where like, you know, it looks like staphylococcus, right? It's very like it's always the same wrong answer.
Yeah.
I don't know if you have any insight into like why or like how would you be able to correct for that? Because it kind of seems like, yeah, lactobacillus would be really hard to accurately correct.
Yeah, I don't know. I was thinking about this yesterday because there is such a big spread, and it's like the D0 is really different than the D8. And so it might be interesting to run the Metacal estimate bias function on just like each sample at a time to see if the bias estimations change for that. But I don't know if it's taking in like if it's just averaging all of them to create the bias estimate or if it's weighting it somehow. But that, yeah, it's definitely interesting to think about, and I'd have to do more research to understand it.
Thank you.
Okay, well, if we have no further questions, I too want to thank Lily and Palmer for their talks and Lisa for hosting them over the summer. And we'll adjourn at this point. Have a great day, everyone.